Hi everyone, our topic for today is mutation. Let us go over our objectives before we define what mutation is.
Objectives.
Number 1, define gene mutation. Number 2, describe the symptoms and mutations involved in sickle cell anemia. 3, state that mutation is the way in which new alleles are formed. 4, use genetic diagrams to show how sickle cell anemia is inherited. 5. State that ionizing radiation and some chemicals increase the rate of mutation. And 6. Explain the distribution of sickle cell allele with reference to the distribution of malaria. So, what is mutation? Mutation is the change in the base sequence of the DNA. And to know more how this happens, let us take a look in this video.
A gene is a bit like a jigsaw puzzle. It fits together like this.
A full strand of the double helix forms a colored pattern. But the other strand always pairs up the same way. A blue base always goes with yellow, and green always goes with red. Because only those colors have the right shape to fit together.
What Crick and Watson realized was that this provides a mechanism for passing on the genetic code.
When cells reproduce, the two strands of DNA separate, ready to be copied. But red still goes with green, and yellow still goes with blue. So, bit by bit, the cell creates two new strands. Two perfect copies of the entire genetic code.
So far, there's no genetic variation. This new copy is identical to the original. But here's the interesting bit. During the copying process, something very important can happen.
Sometimes mistakes creep in. They're called mutations.
Let's have a look at these two bases here. The two prongs that hold them together are subatomic particles. They're protons. They're basically the bonds between the strands of DNA. These protons can jump across to the other side. If the strands split when the protons have jumped across, they find themselves in the wrong position. Now, this red base will no longer bind to a green base. Instead, it has to bond to a yellow base. Slotting this back in, we see that now this copy is no longer identical to the original because I have a yellow base here instead of a green one. We've brought in a genetic mutation.
Mutation in our cells have three different types. First one is substitution. Second, deletion. And the third one is insertion. Let us first take a look what substitution is. Substitution is the most common type of mutation, and this one is also known as point mutation. For example, let us take this as our normal sequence of DNA for a certain protein, G, C, T, A, T, A, C, G. But if you would look at the mutant one, your letter A changed into C, or adenine into cytosine. And this one will cause or produce new proteins to be manufactured by your ribosomes, and that could affect different metabolic activities within the cell. Number 2, we have deletion. From the name itself, there is something deleted, or to be more precise, there is one or more base pairs that are lost, and this also may result in a defective protein. For example, you have GCTA-TAGC as your normal DNA base, but in the mutant version wherein deletion happened, this letter A in here became missing in this certain mutant DNA sequence. Therefore, this will result in a defective protein. And last but not the least, we have insertion. From the name itself, there is something added to the base pair sequence, and this can have a different variety of outcome. One, there could be a similar protein can be produced, or two, a different protein will be created after the base sequence is used in protein synthesis. For example, we have here the normal ones, but the mutant one will have an extra or a new C or cytosine. So where do mutation come from, or what are the different causes of mutation that is happening in our body? First, we have chemical damage. This includes tar, which is found in tobacco. It has different carcinogenic substances in it, over 1,000. And then you have different drugs and known carcinogens. Second would be errors in replication found in the DNA, which is actually used for protein synthesis. Third, you have ionizing radiation, which includes UV rays, X rays, alpha rays, beta rays, and gamma rays. All of these radiation involved or listed different have different penetration levels. And last but not the least, we have viral mutation. The most famous example of virus that mutate our body would be HIV, wherein the material of the human immunodeficiency virus gets to combine with the DNA of humans. And from there, they are able to create or infect the white blood cells that are involved in HIV infections, which will further lead to AIDS. Now, going back to an example of a mutation, this image you are seeing on your left is an example of a condition known as sickle cell anemia. This one in here, this cell in here, these circular ones are all examples of a normal red blood cell. But this one in here, this elongated shapes, very tapered at the side, long body. These are example of a red blood cell that has turned into a sickle cell. Okay, there is a mutation that is happening in the hemoglobin, and this mutation deforms your red blood cell from a biconcave shape, biconcave, to a sickle shape. Okay, this sickle shape is taken from that farming tool that they use in harvesting rice. What happens when your sickle cell becomes present in the system is that they actually accumulate in this narrow portion of the vessels, and this causes different blockages. And if there is a blockage of your red blood cell that is essential for oxygen, then there will be a shortage of oxygen distribution in different parts of the body. But what is difficult with managing sickle cells is that they are stiff and very inflexible, unlike your regular red blood cells, which are very flexible and able to not really change in shape, but able to fold if there are very thin spaces like that or like this in here. This is the mutation that is happening in your red blood cell. So if your usual code or DNA sequence would be this CCUGAG, what happens is that in this second code or group of codes, or also known as triplet, this A in here changes into letter U, which is a uracil, further changing the protein or amino acid, into glue, and then change into valine. Rather than having a hemoglobin with four components, you have now a protein or hemoglobin with
1, 2, 3, 4, 5, 6 times 2, 12.
so it becomes longer, and this is the reason why it is stiffer. How do we inherit sickle cell anemia? Sickle cell anemia would only be found in a person if they are able to inherit both parents' allele for sickle cell anemia. Okay, so let us first take a look at this first Punnett square on your left. So HbA is the symbol for normal allele. HbS is the symbol for sickle cell allele. So if your mother is a carrier, she will have the genotype HbA and HbS, meaning she is normal, but her recessive gene for sickle cell is present. And then for the father, he would have to have the similar genotype as well. Once you have done your Punnett square, you would be getting HbA, which is for normal. You have HbA, which is a carrier, another carrier, and an offspring that is suffering from sickle cell anemia. You would have to have both sickle cell anemia allele in your genotype so that it can be expressed as the phenotype of sickle cell anemia. So if you have a carrier mother and carrier father, there is 25% for an offspring to be normal. You have 50% chances of being a carrier, and another 25% if you are affected by this condition. But what if you have a normal father and you have a carrier mother? So in here, the same genotype would follow. Carrier mother will be HbA, HbS. Father will be normal with HbA, HbA. Apply your Punnett square, and the result will be 50% normal offspring and 50% carrier offspring, but no one will be affected by sickle cell anemia. Malaria and sickle cell anemia. Malaria is a serious and sometimes fatal disease caused by a parasite that is commonly found within a mosquito. Mosquito feeds on humans, which will further spread the parasite into the human body. Once it's inside the human body, the parasite will be invading the red blood cells and they will be reproducing inside them. A person who gets malaria are typically very sick with high fevers, shaking chills, and flu-like sickness. There are 4 different kinds of malaria parasites. This includes Plasmodium falciparum, Plasmodium vivax, Plasmodium ovale, and Plasmodium malariae. There is another one known as Plasmodium knowlesi, which is a type of malaria that can be found in macaques, which are a group of monkeys or primates found in Southeast Asia. It is unique because they do not just affect humans, they also affect macaques, which are a type of a monkey. I'll show an image somewhere here, there. So every time there will be rainy season, make sure your house or your household is prepared by ensuring that there are no places with stagnant water where your mosquitoes can increase in number. And try to use anti-mosquito lotion or insecticides, citronella. Also, that works as well in order to prevent mosquitoes from biting you, because we do not know if they carry parasite for plasmodium or for malaria. They can also cause dengue, they can cause yellow fever as well. So as much as possible, let's decrease the exposure of ourselves to these nasty insects that can carry different viruses and parasites. So what's up with malaria and sickle cell anemia? For comparison, in United States, about 1 in 500 African Americans develop sickle cell anemia. In Africa, 1 in 100 individuals develop the disease. So we could say there is a higher frequency of occurrence in Africa, where there are many endemic mosquitoes that carry the different parasites in their system. And there is a suggested correlation between the population within the African populations, malaria and sickle cell disease. According to the theory, if a person who is heterozygous for sickle cell anemia, also known as this, they are protected from malaria, and the negative effects of sickle cell allele are only present in a small proportion of people who are homozygous for the condition or for the allele. So that includes HbA, and HbS. So if you are normal, and if you are suffering from sickle cell, then there is a high chance for you to get malaria. But if you are just a carrier, heterozygous for sickle cell, then most likely you have little or less chances of getting infected. We could also show this one through this diagram in here. In the left, you have sickle cell allele occurrence in Africa. I'm not really sure when was this data, but let's just take it as a theoretical situation. And then on your right, you have the occurrence of malaria in Africa. If you would notice, this green area in your left are also the ones who are experiencing the malaria situation on the right.